quick recapitulation. In our last class, we were discussing about the translation. We said that we know that translation is the process where mRNA is converted into the proteins. And we said that translation takes place in the cytoplasm. And then we said that these are the following steps for the translation, like activation, initiation, elongation, and the termination. In that class, we had finished activation, right? We had finished activation and charging of tRNA. We had finished in the last class. If you remember, we spoke about how your amino acid combines with the ATP and it gets activated. And then we said that once the amino acid is activated, it's going to charge the tRNA. What does that mean? That means amino acid is going to get attached to the 3' end. Remember this point, to the 3' end of the tRNA, which has a free 3' OH end. Okay, that's what we'll, and then we said that this is called as the, and this is catalyzed by a special kind of enzyme called as aminoacyl tRNA synthetase. And we said in the last class that we have 20 amino acids, we have 20 in specific enzymes for those 20 transfer RNAs, but we don't have only 20 tRNAs, we have more than 45 tRNA, like 45 to 50 tRNAs are there. Okay, that's what we learned in the previous class. And then we also spoke about how important it is to make sure that the right amino acid is attached to the 3' end of the tRNA. Okay, so now today we are going to discuss the second step, that would be this step. Let me show you this step. Today we'll be discussing initiation, elongation, and termination. Okay, so we start with the initiation. So I want all of you to pay attention what happens in the initiation. Okay, so initiation, we're looking forward to form a 30x initiation complex. What does that mean? That means, okay, usually when the protein synthesis has to start, okay, once the proteins have been synthesized, the two subunits, okay, we know that the prokaryotes have 70s subunit, the altogether one, and eukaryotes have 80s, right? But here we'll discuss about the prokaryotic one. So that means 70s has a one smaller subunit and one larger subunit. So smaller one would be the larger one will be the 50s, okay, and the smaller one will be the 30s. Okay, S is the unit given to it based on its sedimentation rate. Savardberg unit or a sedimentation constant, we call it. Okay. Now this is a smaller unit. This is the larger unit of the prokaryotic. We are talking about the prokaryotic, initiation of the translation of the translation process. Okay, we are not talking about the eukaryotic one. Okay, remember that. Okay, so we know that the first step is basically the formation of the pre-initiation complex. What does that mean? That means, as you can see here, this 30s subunit, okay, these are dissociated. The moment protein synthesis is over, it's finished, right? So they separate from each other. So 50s will be lying somewhere else and 30s will be lying somewhere else in the cytoplasm. Okay, and what's going to happen, the 30, as you can see here, this 30th subunit along with certain proteins, okay, called as initiation factors. IF is the word for the initiation factor. IF1, IF2, and IF3 we have here. Okay, so the step one will be the smaller subunit of the ribosome called as a 30th subunit. It will bind to the mRNA. As you can see here, the green one is the mRNA. Okay, along with these initiation factors and along with the some amount of energy is being supplied. Okay, so what's going to happen? Okay, what is Shine-Dalgarno sequence? Okay, so the way we put, for example, you can, the way we have, for example, we eat a burger, right? And we have a bigger subunit and a smaller subunit, right? So before you could assemble the burger, okay, for example, you just take one part of the burger and you slip on the cheese on it, right? The way you put cheese on it or a tomato slice on it, similarly, the mRNA is going to slip on to this smaller subunit. It can slide through the smaller subunit. Okay, and it's going to slide, and this 30th subunit, it's going to interact in such a way, and if you know that the translation will start at this initiation codon AUG, right? So this ribosome is going to screen this mRNA till it finds this AUG. What is this AUG? It is a start codon. Along with that, it's going to interact with the initiation factors. Are we okay till now? Did you all understand till now? Is it okay? Okay. Now you might think that what is the Shine-Delgado sequence? We never heard about it. Shine-Delgado sequence is basically a special kind of base sequence, like maybe adenine, thymine, guanine, cytosine, which is present on the mRNA of the bacterial mRNA, right? So this Shine-Delgado sequence is a sequence present on the what mRNA of the bacteria. Now you might think that why is it important? Why is it shown over here? This sequence is so very important because this mRNA has to bind to the smaller subunit, right? In order to bind with the smaller subunit, this Shine-Delgado sequence is very important because it helps to recruit the ribosome. It helps to bind to the ribosome, the smaller subunit of ribosome. Okay, so binding of mRNA to the smaller subunit of ribosome, right, is basically taken care by the Shine-Delgado sequence. You can say this sequence is very much required, okay, to start the protein synthesis. Okay, it helps aligning the ribosome with the mRNA. Now, how is it possible? Let's say, for example, whatever sequence we have here, okay, let's say we have here uracil, guanine, uracil, whatever it is. So the opposite will be exactly present within the ribosome, right? Opposite will be present within the ribosome. So once these two complementary sequences come together, so that's basically, okay, now the smaller subunit is bound to the, what? It's bound to the mRNA. So this is called as a Shine-Delgado sequence. So can I say that Shine-Delgado sequence is very important for the binding of the mRNA to the smaller subunit of ribosome? Can I say that? In bacteria, I repeat, in bacteria, we don't have Shine-Delgado sequence in the eukaryotes. Okay, yes. So now, even though it's not there in the book, but I just want to brief you about how does this happen in the eukaryotes? How do eukaryotes select their start codon? Okay, so in eukaryotes, if you remember, we spoke about we have post-transcriptional modification. We have a 5' end. If you remember, we spoke about 5' and a 3' end, right? Both the ends are capped. We have a 7-methyl guanosine here in the eukaryotic mRNA, and we have a poly A tailing here, AAA, right? We discussed that previously in the transcription, right? So this, for the eukaryotes, this 5' end is the one which is basically, which helps in identifying the, okay, I need to bind to this ribosome, okay? And then the scanning starts in the 3' direction, and the ribosome stops till it finds the AUG. So it will be, kind of screened in the eukaryotes. It's kind of screened. Okay, then, and then as we have the AUG, okay, now this is the start of the translation. Okay, and then we also have one more sequence. The way we have a Shine-Delgado sequence here, there we have a Kozak sequence, but no need to know all that. Okay, are we okay till now? Okay, Yomna, no problem there. No issues, no issues, no issues. Okay. So till now, we just learned that how the smaller subunit of the bacterial ribosome binds to the mRNA. Okay. At the same time, that look at this. This is a tRNA which has been charged, isn't it? This is tRNA. This is it's what anticodon sequence. Okay, it is also binding to one of the initiation factors, IF2. And this is your charged tRNA. Now everyone will pay attention here. Okay. So we spoke about the previous step that amino acid binds to the 3' end of the tRNA, right? Yes or no, ladies? Yes, right? So we have a difference in the prokaryotes and the eukaryotes. Okay, in eukaryotes, like you have to remember this, that the amino acid which binds to this 3' end is always a methionine. Remember, it's the methionine, right? But in prokaryotes, okay, it is called as it's the methionine again, but there's a little bit of changes, in the methionine. So we call it's the tRNA F methionine. So we call it F, that's how we write it, F methionine. Okay. So in short, F is for the formyl, formyl methionine. Okay. Some chemical changes will take place in the methionine and forms the formyl methionine. Okay. So all you need to remember is that in eukaryotes, the first amino acid, okay, which binds to the tRNA, okay, is always methionine, while as in prokaryotes, it's the formyl methionine. So we will have here the formyl methionine in the prokaryotes. Are we okay till now? Did you all understand? Okay, so the question says that name the special tRNA which starts the translation in eukaryotes. What will you say? Name that special unique tRNA which starts the translation. So what will you say? Which one is that? In eukaryotes, what's that unique tRNA called? Nobody? Where is Muhammad, Dana, Faiza, Hana, Tasneem? No, Tasneem is not here. Tashil. It's the methionine, absolutely. It's the methionine in, absolutely. Thank you, Muhammad and Aileen. It's methionine in eukaryotes, while as it is the tRNA F methionine in D. So don't remember that the way we write it. So we write that tRNA, that special unique tRNA is written like this. So if you see a question and you see that, okay, this tRNA has F methionine at the top. So you have to know that this is the unique tRNA which starts the translation. For eukaryotes, okay, you have to know for the eukaryotes as well, it is the methionine. That's how we write the tRNA methionine. That special tRNA, unique tRNA is called, right? Which carries the formylated methionine in the prokaryotes, while as in the eukaryotes, just the methionine. Okay, so all this, so we have now mRNA, we have the initiation factors, we have the 30S subunit, and we have the tRNA. Look at this. This is that unique tRNA which has the anticodon arm here and which has the special amino acid here and formyl methionine attached to it, right? So all these things will form the 30S initiation complex. Sometimes there's a question which says that name all the components of the initiation complex. So what are the components of initiation complex? We have the 30S subunit, we have the mRNA, and we have the special kind of unique tRNA called as the tRNA F methionine. Along with that, we also have some energy provided by the GTP, and we also have, don't forget the initiation factors, these poor people, right? We also need them for the initiation complex, IF1, IF2, IF3, right? And as soon as the tRNA attaches to it, The first step, what the first thing what tRNA is going to do, is going to find out where is my complementary sequence, where is my codon. Remember, UAC is a codon, anti-codon for AUG. So it's going to screen the mRNA and look at this. Like both of them have, have attached to each other. Are we okay? Everybody? Just like to show you the two ribosomes of the eukaryotes and prokaryotes. So you understood the initiation complex. Okay, great. Wonderful. Okay. Now the question is, so that was all about the initiation complex. Okay. Now, once we have what is the final step of this initiation complex? The final step of this initiation complex would be that, okay, we have to add the larger subunit. This poor person has to be added. Like you made a burger for yourself. You took a smaller subunit, you put all your patty, cheese, everything on the burger, okay? And now you need to bring the bigger subunit to cover your burger, right? That completes your burger, right? So same thing, now we need a 50S subunit to complete this initiation step, okay? Now, before we discuss about the further about elongation, we need to know the structure of the internal structure of the 50S ribosome. Even though I'm sure girls know about it because we discussed that in chapter 3 last semester. I'm not sure whether boys know this or not. So I'm going to anyways repeat it. Okay, it's there in your book, so you have to know it. Okay. So now, if you see here, the larger subunit of the ribosome has three places, has three empty slots. Okay, you can say it has three shelves, three slots. So everyone, please pay attention. Like this is the basics. Then only you will be able to understand the elongation and the other steps of the translation. Okay. So what are these three slots? I've brought it here so that they look a little bigger than the diagram which is in your book. So look at this. This is one, this is two, and this is three slot. There are three slots present on the larger subunit. If it's a prokaryotic subunit, then it's a 50S. If it's a eukaryotic, then it's the 60S subunit. So one is called as an E site, another one is called as a P site, third one is called as an A site. Okay, E site is like a Karuj, exit site. People moving out of the area, right? So tRNA is going out of the larger subunit, will finally exit from this E site. Okay. Now, what is this P site and the A site? These two sites are very, very important. Why do I say so? Because these two sites, this is called as a peptidyl site. Okay. So the first tRNA, okay, which is the first tRNA? The first tRNA amino acid complex, which is that one?
69.
It's the, once again, sorry, I didn't hear the voice.
Methionine.
Yes, methionine is the amino acid. You're right, absolutely. But you have to say it's tRNA methionine for the eukaryotes, right? But here, as you said that, as I told you that in your book, at certain places they're talking about translation in bacteria. At certain places they're talking about translation, in eukaryotes. So it's better both of them, right? So formyl methionine is for the bacteria. So we're going to talk about formyl methionine here. So it's the tRNA F methionine, right? So the first, that special unique tRNA, that this tRNA, okay, the unique one, tRNA F methionine, okay, this tRNA, this one, this tRNA is going to sit at this place, the center place called as the, what is this called? As the P site or the peptidyl site. A very important point to note down here. Okay, this is the only tRNA which enters into the ribosome through the P site. Rest all the tRNAs, you will see that they will enter through this A site, which is called as an acceptor or the amino acyl site. So did you all understand? So that means your first special tRNA with the F-methionine, which has the first amino acid, is going to fill this place. Remember this point, okay, everyone. Okay, that is a very important. So that is a very important point to note down here. Okay, and the second thing what you have to note down here about the P site is that P site is the site which actually holds the tRNA where the long polypeptide chain is growing. Like I will have one amino acid, then another one, then another one, then another one, then another one. Okay, so the tRNA which actually holds that growing polypeptide chain stays here in the peptidyl site. So 2 points you need to remember for the peptidyl site. One, this is the only site where your special methionine, formal methionine tRNA occupies. Two, right, it holds the tRNA molecule wherein the long polypeptide chain is attached. These 2 points you need to note about the peptidyl site. Are we okay? Then what is A site for? A site is like, A site is like the one, for example, if you have a guest coming at your home, right? So you will open the main door, right? So they all will enter from the main door, right? So think that A site is that main door, right? All your guests, all your tRNAs, which are charged with the amino acid, okay, they will all occupy the A site. Are we okay till now? So all the new incoming TR. So only one TRN is a special one, You don't allow that person to enter in through the A site. You simply, I don't know how you manage that. You simply take that, let's say P is like your, the main room where all guests are to be, they have to sit. Okay. You simply take the first TRN into this main room. Okay. You don't allow that TRN to go through the A1 and then come into the P1, right? So that is an exception which you have to remember. Rest all the tRNAs, all 19 tRNAs are going to enter through the A site, which is called as an amino acyl site or the A site or just the acceptor site. Are we okay about all three sites? And the E I told you is like a Kharuj one, where everybody will leave. Yalla, go, bye. Thank you for coming. Okay? Are we okay till now, guys and girls? This is like the
Yes,
most
doctor.
complicated thing, Yes, okay. So you understood the three sites now, right? Okay. Look at this, what is happening. Now, once we have the larger subunit attached to this initiation complex, okay, what's going to happen, as I just now told you that this is my special unique tRNA. Look at this. This poor person was not allowed to enter through A site. He simply sat in the B site, the main area where all the guests have to sit. Okay. Okay. So now you will see that slowly and slowly this completely forms the initiation complex, and now we have everything. We have the 30th subunit, we have the mRNA, we have the special first tRNA, we have the larger subunit, right? We are all okay now. We are ready to start the protein synthesis now. Okay, everything is in place, and now we'll do with the what so this is like, I put this slide here so that you don't forget this. Okay, whatever I said right now. Okay, so now we'll move on to the elongation. Okay, we need to elongate the chain now. We need to keep on adding the amino acids. Okay, now in order to add the amino acids, what's going to happen here? What do you think? What should be the step here? What should happen now? Okay, ladies, what's happening here? tRNA. No, now Okay, what's happening in the elongation? In the elongation, okay, first I had this first amino acid, right? Then the next tRNA will come, it will also bring amino acid, right? So this chain, which had just the F-methionine, you will see that this chain will slowly and slowly become longer and longer and longer and longer and longer. That is the main aim of elongation. Okay, that is the main aim of elongation, right? And I forgot to tell you about here, okay, when the anticodon of the tRNA, it's fixed to the codon of this, mRNA, okay, these, initiation factors, okay, they all are released from the initiation complex. You don't see an initiation factor over here, okay. We were in need of them only till here. Now we don't need them, okay, and not even the energy, because the energy has been used, okay, and now we only have all these four components in the form now, okay, in the initiation of the translation. So now now the next step is that we need to bring more and more tRNAs, because we know that more and more tRNAs will bring more and more amino acids based on what sequence we have here, okay. Based on that, the incoming tRNAs will be recruited, new guests will be recruited based on what message is present or what sequence we have on the mRNA. That's going to happen in the elongation. Are we okay till now, everybody? Any question till now? Now let's do how that happens. Okay, so now you'll see, as you see, this was my first tRNA, okay. As you see here, you don't have to know all this, okay. As you see here, if you see the next tRNA, it is carrying an amino acid called as an alanine. So it should come, as I told you, the second tRNA and the third and fourth and fifth, they all will come through this A site, right? Look at this. The second tRNA, which is a charged tRNA based on GCU, was a sequence on the mRNA. Accordingly, the tRNA, which had the opposite sequence at CGA on its anticodon arm, was selected and brought into the A site, right? So the second step would be the newly charged tRNA based on what codon we have on the mRNA will enter through the A site. That will be the next step of the elongation. And what do we need here? You will see that, they're all elongation factors. Excuse me, they're all elongation factors, okay? Because the way we were in need of initiation factors earlier, okay, and now we need the elongation factors, okay. So they're all elongation factors, okay, and that is your first step of the elongation. That means a newly charged tRNA is going to sit into the A site, as you can see here. Now, what is the step 2? The step 2 in elongation is now these two amino acids are there. What should happen between them? We should have which bond is present between the two amino acids? The question was there in
Peptide bond.
the midterm, right? Right, I put that question in the midterm. So that was a peptide bond. Very good, right? So the peptide bond is going to be formed there. And of course, we need an enzyme to catalyze the reaction. And the enzyme will be the peptidyl transferase. Its main job is to form the peptide bond between the two adjacent amino acid. Which two? One present in the A site, another one present in the P site. Okay, all clear? Now the bond is formed, khalas. Now what's going to happen? Step 3 is the translocation. What is translocation? That, now the ribosome is going to move. Okay, so ribosome is going to move in the 3' direction. That is the translocation, like towards this side. This is a 3' direction. Okay, now as the ribosome is going to translocate, what do you think? What should happen now? And this is catalyzed with the enzyme called as the translocase. What should happen? As the ribosome moves down one codon to the right, obviously the two tRNAs which were there at the which site? at the P and at the A and the P side. Now the one which has, this one which is now uncharged, it has moved to the E side, the exit side. And the one which was at the A side is going to move at the P side now. Did you all understand? So the two tRNAs, one which was in P side and the A side, are now moved to the E side and the P side because it's the ribosome. Remember, the mRNA does not move. It's the ribosome. mRNA is staying. The cheese is staying wherever it is. It's the two buns which are going to slide over the cheese, if you, just in case if you don't want to have cheese in the burgers, which would never have happened. Is it clear? So this might be a question, for example, is it the mRNA which translocates? No, it's the ribosome which shows. The translocation means movement, moving on, changing the position. Okay, so now my tRNA, which is now the empty tRNA, or which doesn't have anything, it has done its job. It's ready to go out. It will keep on, it will go out from the exit side. The empty tRNA is released from the E side, right? And now what's going to happen after this? Now the third tRNA will go and bind to this side. Again, a polypeptide chain will, the peptide bond will be formed between this and this, right? Then this tRNA will move, right? So the whole process is going to repeat and repeat and repeat and repeat. The elongation is continuously going to happen, okay, till, okay, this is the peptide bond formation, okay. No need to know about the nucleophilic attacks here. Just remember that the peptide bond is formed here, right? Okay. Now what happens in the, so what's going to happen? So look at this. I just started with maybe the methionine. I just had the methionine. Look at this. This is my longer polypeptide chain. All these things will continue unless and until, what's going to happen? How is all these things going to stop? What do you think?
A
How will
stop
it
codon.
Codon.
Yes. Do you remember the 3 stop codons? UAA, UGA, and UAG. Remember these codons, right? Remember these 3 guys. Okay. So these 3 guys are going, the moment any of them, for example, if I have UAA here, or it could be UGA or UAG, right? The moment this is encountered in the mRNA, that gives a message to the tRNA that, okay, khalas, we're done. We need to stop the protein synthesis. Because for these codons, do we have a tRNA for these codons? Do we have an anticodon for these codons? No. There is no tRNA which can bind to your UAA, UGA, or UAG. There's no anticodon, of course not. No anticodon for these 3. So what do we have for these 3? Instead of that, we have, a special kind of a release factor. You can say a special kind of protein. Okay. Once you have UAA, look at this, right? These are the release factors, RF1, RF3, along with we have energy. So they bind to the A side, not the tRNA, right? So thus the A site is filled with our release factor and with some amount of the energy as in the form of a GTP, right? So now the amino acid is, once this will happen, now this is a message. Now you need to cleave the amino acid, right? So amino acid will be cleaved and now it is what? It's free from all these complexes, because it's the release factor over here which will hydrolyze here the polypeptide chain. Okay, and now this is my newly formed protein chain. Okay, and now what's going to happen with the ribosomes? Are they going to be joined together again with the mRNA? What would be the last step in the termination? Are they going to stay back together, the 50S and the 30S in the prokaryotes, or 60S and 40S in the eukaryotes? Are they going to stay back together? No, I told you, the moment your protein is formed, khalas, your two buns of the burgers are going to be separated away, right? They're going to separate again to start the new round of protein synthesis. So all these things will happen repeatedly, and that's how the proteins are being synthesized. Is it clear to everyone? Did you remember last week I showed you a video for this? Do you want to see that video once again, or you're okay? You saw that last week. It's clear. Anyways, I'm going to upload those videos on the Moodle as well. Okay. Any questions about it? Are we okay? No. Okay. So yes, there's a video. So now after comprehending all this, it's better to watch a video. Okay, you can watch right after the class once again, as I will upload it on the Moodle. Okay, and the same video we saw that last week. Okay, so whatever I explained, you will see all these things happening in the video, and it's going to stay there for a longer time. Look at this. Okay, this is my polypeptide chain, and everything has separated now. Okay, look at this. Even the tRNA, is this tRNA a charged tRNA now? No, this is an uncharged tRNA. It will go back again and start all the process of charging with them, with the amino acid again, and start the new synthesis of the protein synthesis. Okay. A quick question. The genetic code is considered degenerate. We discussed about a genetic code last week. Why is it called as degenerate, everyone? We discussed that last week. If you remember, I told you, the word degenerate is very important. You need to know what is the meaning of the word degenerate. What does that mean? Yes. Yes, Lena, Munira, Mustafa Hilal, Rand, Soumya, Tanya, where are you all? Yusuf. Why do we say that genetic code is degenerate? Answer is B. No ways. DNA is unstable.
It means that more than one codon can code for the same amino acid.
Absolutely. Who was that? Is it Jida?
Right.
Right. Thank you, Guide. Thank you. Thank you. Thank you, dear. Absolutely. If you remember, I explained to you that day, when you encounter the word degenerate, it means that more than one triplet we have for the same amino acid, right? We saw that, if you remember, we saw that serine has 6 codons. There are 6 codons which can code just one amino acid called as the serine, right? Similarly, for certain amino acid, you will have 4 codons, you might have 2 codons. So that part is called as the what? Degeneracy of the genetic code. Who will answer this? Do I need to pick and choose? Yomna. Yes, dear. What is the answer for question number 2? How many codons we have all together? C. Yes, Mohammed. Yes. So altogether we have 64 codons, isn't it? Out of that, we have 61 codons which code only for the amino acids. And rest 3 are the nonsense codons called as the stop signals. Very good. Okay. So where is the codon found? An easy one. Who will answer this? Tashil, do you want to answer? Where is the codon found? Codon is found on the mRNA. Yes. A triplet, right? 3 bases together forms 1 codon. Where is anticodon found? Soumya? tRNA, right? The anticodon arm. Very good. So what are codons? Very good. Yes. What are codons? Three bases together on the mRNA forms a codon, which codes for a particular amino acid. Okay. Wonderful. Now, gene regulation. Okay. Genes can be regulated at the transcriptional level. There might be certain changes which can happen in the genes at the transcription level, or sometimes at the translational level as well. Okay. If you remember, I explained you that day about all these, but I'll just do a quick recapitulation because that was the last week. I told you that what are transcription factors? We said that transcription factors are always only needed in the eukaryotes because DNA polymerase has less affinity towards the DNA. It needs a helper protein, and that helper protein is the transcription factor. And if you remember, I told you that the transcription factors are called as, they help in they're very important in the regulation of the transcription. Okay. So if you remember, in this diagram we said that once the promoter is identified by the transcription factors, then only the RNA polymerase is recruited, or then only it binds to this whole assembly. Okay. So we also spoke about that the promoter called the Tata box is recognized by the transcription factors called as the Tata binding protein. We discussed all that last semester. Then here is telling you that, and we also spoke about the enhancers, silencers. A quick recapitulation, we did all this last week. Enhancers, we said that it is also a group of transcription factors which will simply speed up the transcription. Okay, they're called enhancers. Okay. So if you remember, I told you when the activators bind to these enhancers, right? So the transcription rate is going to increase. Okay. And then the silencers, we also spoke about silencers, right? Silencers, we said that silencers are basically those types of proteins, okay, which slow down the transcription, right? We sometimes call them as the repressors or just the silencers. Okay, the moment they bind to the DNA sequences, they inhibit the start of the transcription, right? So transcription is stopped the moment you have your the silencers attached to the DNA, right? That's what we learned last week, I mean, right? And then I also spoke about the response elements. Okay, I told you there are also the short sequences present within the gene promoters. They also are called as regulators of the transcription. So all these help in the regulation of the transcription. Now, the question is, how do these transcription factors find a specific sequence and know that, oh, I want to bind to this? I showed you a diagram here, right? For example, if this is my DNA, and this is my transcription factor TF, for example, 1. How is it possible that this TF1 knows where exactly I need to bind, to which part or which promoter I need to bind? Okay, how is the specificity between the DNA and the transcription factor, right? For that, okay, this is like something, it's not explained in detail in your book, so I will also not go into details, but whatever is there in the book, let's try to learn that, know that, I mean. Okay, so any transcription factor, whenever you see any transcription factor, it has, very, lot of protein domains, okay, you can say the various parts, okay, with special type of amino acids, which can actually have, which has, attraction towards the DNA. They interact with that DNA. Okay, so if you see here, what kind of structure, what kind of protein structure is this? Is it primary, secondary, tertiary? This one, this helix, what is it?
Secondary.
Yes, right, it's the alpha helix, isn't it? Yes. Okay, so yeah, So what's happening basically? So there is something called as the zinc finger motifs. Okay, we also call them as the metal binding fingers. Okay, so what are these zinc finger motifs? Okay, there we have special amino acids like your, for example, your zinc, which is attached to the cysteine as well as the histidine, as you can see over here. So whenever you have these kind of sequences present in the transcription factor, okay, these sequences promote the binding of a transcription factor to the DNA. Okay, did you all understand what I'm trying to say? Right? So it's all about the side chains of these amino acids, which will interact with the, with the DNA. Okay, that's how they identify their DNA. Okay, do you all understand? If you see here, this has formed a loop. Look at this, has formed a loop. Okay, wherein you have two cysteines, this cysteine and this cysteine. It might form a bond here, right? There was a question also yesterday, which bond will be formed over here? Can anyone tell me? Between these two cysteines?
Disulfide bond.
There was a question also in the midterm. Yeah, disulfide bond will be formed here, right? This is so special. This is a specialty about the side chain of the cysteine amino acid, right? So there will be a disulfide bond formed here, bringing these two domains close to each other, right? And then this cysteine is further connected with the zinc, okay? And this zinc is further connected with these two histidines, okay? And it forms, a finger-like structure. Do you see a finger-like structure here, which can clasp, to the DNA? I will show you the next diagram. Look at this. It has formed this, finger-like structure, okay? And this finger-like structure, it kind of, it holds the DNA, okay? And thanks to these amino acids as well as the zinc, because of their presence and because of their special side chains, your transcription factor are able to attach to the DNA, right? So zinc fingers are very important. Now, if you say that what is the function of a zinc finger, you will say that zinc finger, the main function of zinc finger is that it simply plays an important role in binding of the DNA during transcription, because these transcription factors need to bind to the DNA. Are we okay about it? So you can say that they are their DNA binding domains, okay, which have, more attraction towards binding the DNA. Is it clear? Any questions about it? Okay, so in your book you only have zinc fingers, but we also have many other domains like helix turn helix. Okay, there are also um, you know some domains present in the transcription factors, okay, which help in the binding of transcription factors to the DNA. What about others? Is it clear to everyone?
Yes, doctor, clear.
Okay, dear, thank you. Okay, now other than that, we'll also talk about now the gene regulation. Okay, so Human Genome Project, which was started in 1990 with so many countries and so many expertise people in the world, their main aim was to sequence the human DNA, and which they actually achieved successfully, right? So I will not go into Human Genome Project, okay, but it was all about sequencing the entire DNA of human beings, okay. So they found that there are almost 30,000 genes present in the humans after this project was completed, okay. And these 30,000 genes form 90,000 different proteins. Do you understand what I'm trying to say? Ideally, we say one gene forms one mRNA. One mRNA forms one protein. So ideally, how many proteins should have these genes made? 30,000 genes?
30,000 proteins.
Absolutely, very good, right? That should have been the real count. We should have only 30,000 proteins. But to their surprise, they said that, oh my God, only 30,000 genes are forming 90,000 proteins. So how is it possible? How can these 30,000 genes form 90,000 protein? The answer they got was the alternate splicing. What is an alternate splicing? Okay. So once my mRNA is formed or gene is formed, or okay, so this gene can be altered, or you can say edited, in several ways. Okay, like I have A, B, C, D, E, F, G, H on a gene, for example. So I can combine A with C, B with C, D with A with G, G with C, right? So there might be various combinations, okay? And just because of those combinations, we call it alternate splicing, okay? It can give rise to many proteins, though the transcript must be just one. So primary transcript, a primary mRNA which is formed can be edited in several different ways to give multiple mRNAs and thus multiple proteins. And this is done by the splicing activity, right? Did you all understand what I'm trying to say? So in short, you have to say that one mRNA, I just have one mRNA, but you will see that in a while it will be cut in such a way, it will be spliced in such a way so that one mRNA can give me many proteins. That's what they found when they were working on the Human Genome Project, how 30,000 genes can form 90,000 different proteins. Are we okay till now? What do we call it? We call it alternate splicing, correct? Yeah. Let's see now how it happens. Let's say this was my primary gene. You know now what are exons and what are introns. Do we need to remove exons or we need to remove introns? Yes, ladies and boys.
Introns.
Introns, right? They should be spliced. Remember the SNPs, the spliceosome? Yes, they should be spliced. Intron, absolutely. Look at this. So this gene, I can cut this portion from the gene. This whole portion will be cut from the gene so that I can have only this exon and this exon together. Isn't it? I got this mRNA. Or I can splice it in a different way. This is also an exon, but sometimes it's kept there in the gene, sometimes it's spliced. So the other way is I don't remove this whole chunk. I just remove this one and this one. What will I have in the gene? In the mRNA, I will have 1 this, 2 this, and 3 this. Isn't it? Is my this and this same now? Even though I got them from the same gene. Are they same now? Boys and girls. Of course not. Did you understand? What's happening here? Initially they replaced, they omitted, or they replaced the whole thing, not replaced, they removed the whole thing. Okay, look at this. So we had only this and this, so it got me this mRNA. Okay, but second time what happened, the gene was spliced in such a way so that this was kept, and this was kept. So I got altogether a different sequence mRNA. So this can form a different protein, this can form a different protein, though there might be some similarity between these two proteins. Clear? So this is called an alternative splicing. Okay, so same mRNA, but it can be spliced in different ways, which can give us different kinds of proteins from the same genes. Okay, and look at this, what is happening in the second one. In the second one, initially, this was spliced out and this was spliced out, the whole thing. So I had this 1, 2, and 3. Look at this. I had this mRNA. But the second time, what did they did? What did they do? They just left this smaller part over here. Okay, but they removed only this part and this part. So I have this one now here. Look at this. So this will be different from the first one. So obviously, this will form a different polypeptide chain, and this will form a different polypeptide chain. Okay, and the same thing is going to be here as well. So it's just kind of showing you all these combinations. All this is called as an alternative splicing. So eukaryotic RNA can be spliced in more than one way, right? So this is a eukaryotic splicing. Do we have splicing reaction taking, does it take place in the prokaryotes as well? There's the post-transcriptional modification, once the mRNA is formed, once the pre-mRNA is formed, okay? And then if you remember, we said that it will be converted into the mature mRNA and then sent to the cytoplasm. Does that happen in prokaryotes? So you have to be ready with all these questions because there's a lot of distinction between prokaryotes and eukaryotes. No, right? If you remember, we spoke about last time that no post-transcriptional modifications in the prokaryotic mRNA. It only takes place with the eukaryotic mRNA. Okay, that was about the alternative splicing. Okay, now translational control. What do you mean by translational control? That means, okay, now you produce the protein. Okay, now your protein is formed. I had a DNA, I formed mRNA from it, then it went to the ribosomes and I formed my polypeptide chain. I have a protein chain now. So is this protein chain ready? Is it ready? Is it ready? Do you think there's no other post-translational modifications which have to take place? Yes or no? No. We in eukaryotic DNA, right? Once we form the proteins are also, you can say that there might be some changes. It needs to be folded to absolutely, Tanya. After studying the protein chapter, right? You know now how important protein folding is, right? How important the other higher structures are to make a protein functional, right? In order to make a functional protein, we need to do few things so that this protein can be ready to be used. What are those things which are to be done? Okay, so in the post-translational control, we have removal of the methionine. As I told you that the first amino acid in a protein, and after studying translation, you know now, the first amino acid in the protein is always methionine, right? But sometimes in the post-translational modifications, this methionine can be removed also. It depends upon what kind of protein it is and what functionality that protein has. Okay, and chaperoning. Who will tell me what are chaperones? We discussed that, we had that in the chapters which were there for the midterm.
They help fold the proteins into their active conformation.
Absolutely, right? If you remember, we had a diagram which looked like this in the protein chapter, right? It looked like this. And within the chaperones, you had what? The unfolded protein first, and soon you saw that this chaperone is going to fold this protein, for example, in the alpha helix, right? Yes. So chaperone, or we call chaperoning. Chaperoning is simply they are the proteins themselves, but they help the other proteins to get folded. Okay, so that they become active, they become functional protein. God forbid if there's any problem with the chaperoning, okay, the misfolded protein can lead to the various diseases. If you remember, I gave you an example that day. Does anyone remember? Which example, when I was teaching you proteins, which example did I give you when we have a misfolded protein? And which disease can be caused if you have that misfolded protein? I gave an example that day. Exactly, Muhammad. Alzheimer's disease. Very good. Yes. Right. Absolutely. Very good. Thank you for remembering that, Muhammad. Right. So it was the Alzheimer's disease. Okay. So the function of the chaperones is simply to fold the newly formed polypeptide chain so that they can become functional. And number two, what if, okay, I have some proteins which are misfolded. What should I do with those proteins? Right? So do you leave them there so that they can produce some mutations, a tafra in the body? No, we can't leave them there. We have another kind of enzymes called as a proteosomes. Okay, proteosomes are the enzymes which have proteolytic activity, protein breaking activity, right? So what if the chaperones failed to fold a particular protein? Then no worries, that's okay. We still have proteases, right? They are going to degrade that misfolded protein. Clear about this slide? Any question about this? Yes, boys, girls?
Clear, doctor.
Thank you, dear. Okay, so now the mutations, it's basically the same thing what we discussed previously. If you remember in previous chapter, we spoke about the nucleotide excision repair and the base excision repair. Okay, so you need to know the definition. What is a mutagen? Mutagen can be any chemical or any radiation, okay, that can cause a base change or a mutation, a tafra in the DNA. Okay, and we this all these things we spoke about it, if you remember when we were doing proofreading of the DNA polymerases, right, the DNA repair mechanisms. Okay, and we discussed all that, right? So we said that god forbid if there's any problem with the proofreading mechanisms, we still have the DNA repair mechanisms, the nucleotide excision repair, which repairs the damaged DNA by replacing the whole nucleotide, right? The whole big chunk of DNA can be removed by this one, nucleotide excision repair. And as the name says that in the base excision repair, we have just the bases which have been deaminated or whatever change it has been. Okay, so that can be removed by the base excision repair. They are just the DNA repair mechanisms, so which can prevent the mutations. Okay, so this is basically the same thing, in the previous, what we studied in the previous chapter. And now recombinant DNA technology, or just the recombinant DNA. How many of you know about this recombinant DNA? What is the recombinant DNA? Yes. Have you ever heard this term before, or what is the meaning of recombinant? No, never heard of it. Okay, no problem. Okay, nobody. Bacteria carry DNA. Okay, which DNA, Mohammed? Bacteria carrying which DNA? Any DNA or its own DNA? Okay, don't worry. Don't worry. Okay. Even though this is basically a very vast recombinant DNA technology itself is, high-tech technology which needs special attention, and within just 1 or 2 minutes you can't explain it, but I'll just try to sum up everything. You should ideally have a chapter. This is what they use for the insulin. Absolutely. Yes. Not only insulin. Oh my God. Plenty of proteins now. Plenty of enzymes now. Okay. And for the growth factors as well. Now, okay, now let's pay attention. What is the recombinant DNA technology? As I said that it's a vast subject. It's a subject itself. The way you have anatomy, physiology, right, pathology, recombinant DNA technology is itself a subject. Okay, so it will be very difficult for me to explain the whole thing in just a couple of minutes. So let me try to sum everything. Okay, so what is the meaning of recombinant DNA? Okay, recombinant DNA is, for example, I got some DNA from the humans. Okay, I got a DNA from a human being. Let's say whatever DNA it is, whatever gene it is. Okay, I combined this DNA, let's say, with the bacterial DNA or any DNA. Let's say I combine, this is a bacterial DNA. Okay, I combined it with the bacterial DNA. What did I get at the end? At the end, I got a combination molecule or a recombinant molecule, okay, wherein I had a bacterial DNA and then I joined them together using a ligase enzyme, and the red one is the, so this whole will be called as a recombinant DNA. So half of my DNA is the plasmid DNA, is the bacterial DNA, and I also have my gene sequence, my DNA sequence in this type of recombinant DNA. So this is called the recombinant, like recombination, combining the two. Do you understand? And then forming one molecule or one DNA. So it will be having a properties of both the gene which we inserted here, right, and the bacteria which has whose DNA which we used, and that bacteria will also have its own properties. So we have used the DNA from two different sources. One, the human being, and second, the bacterial DNA. Are we okay? But that's not all. This this field has expanded so very much. Recently, the vaccines which you're using right now, whether it's an AstraZeneca vaccine or whether it's a Pfizer one, both of them has been produced using the same recombinant DNA technology. So nowadays we are like incomplete. Medicine is incomplete without, if you don't know about recombinant DNA technology, right? Look at this. Within few months itself, the scientists came up with the vaccines, right? And this all happened. Everything was fastened up just because we have nowadays, we can artificially create DNA. Do you know that? If you know the gene sequence, right, you can artificially create the DNA in your lab, or you can combine the two different sequences. Okay. And now the DNA which I used here, the blue one which I used here was the DNA which was from the bacteria. This DNA is called as a plasmid DNA. So one of the basic pillars of the recombinant DNA technology is that we have a plasmid DNA, which is nothing but just a double-stranded DNA present in our bacteria. Like for example, this is my bacteria, okay? Bacteria has its own DNA, right? But then it has a smaller extra DNA, double-stranded DNA, which is called as a plasmid. And this is so independent, this plasmid, because it has its origin site. It can replicate by itself. It's not dependent on DNA for replication. That's called as a plasmid. So you should know the definitions as well. What is a plasmid? So plasmid is an small circular extra double-stranded DNA present in the bacteria, right? And for the recombinant DNA technology, for any cutting and pasting, you also need the enzymes, right? Which can cut this DNA, right? So for cutting and pasting, for not the pasting, for cutting, you have the enzymes called as the restriction endonucleases. So all this is very important. Let's pay attention. What is happening? Okay. I have a diagram here, maybe that can be helpful for you. Okay. This one. This is a diagram from your book. Okay. So in order to make a recombinant DNA, what did I do? I took the bacteria, okay, opened the bacteria, removed its, cell wall, whatever, took the plasmid, okay. Now please pay attention. What did I do? I removed a part of the plasmid. Okay, do you see here? I removed a part of the plasmid. So I kind of, it was a circular plasmid. Okay, I used my restriction endonucleases. I'll talk about them in a while. These molecular scissors, they're called as molecular scissors. That means, the way you cut your paper, similar, they cut the DNA wherever you want to cut the DNA, right? So using these restriction endonuclease enzymes, I cut this plasmid DNA, okay, and then I got my gene of interest, whatever gene I want to, you know, clone, okay, or have exact copies, okay, and I inserted that DNA within this, plasmid, and I put the plasmid back, because this technology becomes only and only useful when the artificially created DNA, this one, can be what? Cloned. What is cloning? Making more copies of it. If you will have just one copy of it, do you think it can be of useful, you can use it? Right? You cannot use it, isn't it? Whatever you're doing, you should have many copies of it, right? So this was the technique which was used, like the first important, molecule which we got using the recombinant DNA technology, the protein which we got was the insulin, right? So before this technology was there, if doctors or if pharmaceutical industries would need the insulin, they will go to the animals, extract their pancreas, take the beta cells, remove the insulin, right? Purify the insulin, and then make the injections out of it, right? But all these things changed when we were able to first time isolate the gene. We found that, okay, this is the gene which forms the insulin. This is the gene which forms the protein insulin, right? Or the hormone insulin, right? When we were able to identify that gene, and then by using the recombinant DNA technology, we were able to create the insulin in the bacteria. Can you believe? Whatever insulin nowadays we're using, it's all created by the recombinant DNA technology. No more killing of animals. It's all created by this process. What is the process? First of all, take the human DNA, isolate the gene which can produce my insulin, right? Which can produce the insulin. Absolutely. Yes, Tanya and Yomna, right? So take the gene from the human, okay, from a human being. Isolate, for example, I took few cells from the human being, right? Isolated the DNA, okay? Which DNA? The DNA which codes for the insulin, okay? And we spoke about insulin, right? In the protein chapter, right? We know the sequence of the insulin. There was a table for the sequence of the insulin, right? So took that insulin, took that DNA which produces the insulin, okay, and then what did we do? We cut the DNA using the restriction enzymes, the molecular scissors, okay. We just wanted this red part because we knew that only this red part produces the insulin. We didn't want other genes. We didn't want all these. Okay, so we cut them away. Okay, now this is my target DNA. I have the insulin gene ready. What do I need to do? I need a bacterial plasmid. I told you now, right? Bacteria has an extra double circular DNA called as a plasmid. Okay, So I took this plasmid away from the bacteria. I also cut this plasmid. Look at this. I cut this plasmid. Okay. And so like if the plasmid was like this, I took the whole chunk away so that I opened the plasmid like this. Now within this opened plasmid, I can fill in my gene of interest. This is my gene of interest, which is insulin, the red one. Look at this. And put the plasmid back in the bacteria, right? Bacteria will become from 1 to 2, 2 to 4, 4 to 8. It will keep on dividing in special kind of fermenters, which they use in the pharmaceutical companies, right? And millions and billions of bacteria are produced, and they're the machines which are producing your what? Insulin hormone. Then it was isolated for the first time, and it was a greatest breakthrough in the entire, medicine. It was considered one of the, greatest breakthrough that you know how many diabetic patients are there, right? So this has made our life so very easy. Now tell me how many of you understood the process, or I will repeat it once again. And did you find it interesting? Everybody, isn't it? Yes. And you will be surprised to hear now. We spoke about interferons. Remember interferons in lymphatic system? Yes. Nowadays, not only insulin, you know about the human growth hormone. We spoke about the chapter number 9. How human growth hormone artificially, if you give it to the patient, right? How they can increase their height. Nowadays, whether it's the insulin, human growth hormone, your interferons, okay, or erythropoietin. Remember the erythropoietin? Yes, if the patient has anemia, you will give them erythropoietin, which is naturally produced by our kidney, chapter number 9. But I'm giving you these examples because you have read about all these things. So insulin, human growth hormone, erythropoietin, interferons, right? Or factor 8 for hemophilia. All these are nowadays produced by the same technology. Many anti-cancer drugs. I just gave you a couple of Taxol. We all know how important Taxol is for the ovarian cancer. Hepatitis B vaccine. And how can we forget the vaccines which we recently used, whether it was a Pfizer or it was the AstraZeneca one, used by the same technology, right? So you can now understand how important this technique is. Being a doctor or being a researcher, you should be knowing about all this, right? These are the techniques which has made our life, which has simplified our life. Okay, now I use the word restriction endonucleases again and again. I told you that, cutting like the scissors, the way we cut the clothes, or we cut the papers, right? We use the scissors, right? So here we use the enzymes called as the restriction endonucleases, like these enzymes. So please pay attention. So what is so special about these enzymes? These enzymes can cut a particular DNA at a particular sequence. Let's say I have a restriction enzyme 1, let's say 1. It is specialized in cutting the DNA only at adenine and guanine. Wherever it finds adenine and guanine, it's going to cut it. Let's see the next slide. Look at this. This is my human gene. This is my bacterial gene. I had GAATTC, CTTAAG, like the complementary sequences. I use my scissor, which is called a restriction endonucleases, right? So I told you that this one is specialized in cutting what? A and G. So wherever it will find A and G, it had A here, right? And G here. So it's going to cut here and here. G here and A here. Look at this. I have now 2 different sequences of DNA. Like I had this DNA, now it is in 2 pieces, and it has these hanging ends. Okay, we call them sticky ends. So these, it gives a staggered cut. You know what's a staggered cut? If you cut a carrot, if you have to cut a carrot, for example, you can cut it like this. So it will be, into two halves. But if you give a diagonal cut like this, so your carrot might, might look like this one half, and the other half might look like this, right? A little slanted cut over here, right? It's called staggered cut. So your restriction endonucleases are, very important in giving these staggered cuts. Okay, and same enzyme we can use for the human gene as well, cutting, and then only we can join them together, right? These enzymes are very, very important, right? Very, very important. Without them, it was impossible to achieve whatever we have achieved till now. That's why we call them, molecular scissors. Okay, now cutting is not only important, joining is also important. You've spliced the plasmid, or you've spliced the human DNA, right? You need to join it back. So of course we have the DNA ligase for joining the things back, for joining the plasmids with the our gene of interest. Is it clear to everyone? So dash are the enzymes also called as DNA scissors or the molecular scissors. So it's the restriction endonuclease. Island says it is a cut at ATC. No, as I said that every enzyme has a different, I just gave an example here, right? For example, we have an enzyme here, which I have also used in the lab. So when I used to use EcoR1 was an enzyme, it was an endonuclease. It used to cut the sequence wherever it finds, I used to remember that sequence now. No, it was always on the tip of my tongue because I use this so often. It used to cut the DNA wherever it finds guanine, adenine, thymine, cytosine, right? So you don't have to remember the sequence here, okay? You just need to remember what are restriction endonucleases, where do we use them, okay? Why, what is their speciality, right? They can degrade the phosphodiester bond, okay? And they're so specialized, we have so many restriction endonucleases, and every enzyme is, they're specialized in cutting, different kinds of DNAs, okay? And they are there to create the sticky ends in the DNA, okay? So two fragments are joined by the DNA ligase enzymes. And inserting DNA fragment of interest into DNA is called as a recombinant DNA technology. The last topic of this chapter is the gene therapy. Have you ever heard about gene therapy? Boys and girls, what is gene therapy? Quickly, I have a class to start. We couldn't. Okay. Gene therapy is simply if a person has to correct the mutations. Yes. If a person has any gene which has gone bad, okay, or any genetic, diseases which we have, right? So we use gene therapy for that, okay. I don't think so we can finish it. So this topic would take just 1 minute, but I have a class to start, okay. It's already 10:28. Yes, that's what I was wondering. I wanted to discuss about the project, but that's okay. We have a Wednesday class. So Wednesday, we can have a combined class, and Wednesday, for the first half an hour, 40 minutes, we'll only talk about the project. Because anyways, you will not start preparing it now because, you just need to submit the topics yet, right? So on Wednesday, there's no other way out. We have to discuss about the project. It's very, very important. Would that be okay, everyone? On Wednesday, we'll not start with the class. We will start with discussing the project, and then we'll start with the class. Would that be okay? Okay, and then the gene therapy is like, we'll discuss within 1 minute in the next class. Okay. Thank you, everyone, and bye-bye. I have a class to catch. Bye-bye, everyone.
Thank you, doctor. Have a nice day.
You too. You too. Bye-bye, everyone.